we've known for a while now that FGFR2 is a reasonable target in esophageal gastric cancer. Through next generation sequencing, it appears that there is amplification of the FGFR2 gene about 5% of the time. It's not high, but certainly it does represent a legitimate target. So the FIGHT study really evaluated targeting this with an antibody called bemarituzumab, which targets the FGFR2 B receptor. And I think what's critical is how common the target is. So their positivity was defined either by immunohistochemistry of tumor tissue looking for the protein, or circulating DNA looking for FGFR2 beta amplification. Ultimately, about 30% of tumors that are HER2 negative were found to be positive by these criteria, although the majority of it really was by protein expression at the IHC level. So in any case, this actually was originally designed as a randomized phase 3 study, but subsequently became a smaller randomized phase 2 study. And it evaluated FOLFOX with or without bemarituzumab for these FGFR2 beta 2B positive tumors. So the study met its primary, which was investigator-assessed progression-free survival, but there was also improvement in overall survival as well as response rate. The experimental drug itself was associated with a higher risk of stomatitis, as well as some corneal, so eye toxicity, but otherwise appeared to be relatively well tolerated. So on the basis of this, the investigators are proposing that a phase 3 study should ultimately be launched. I think that's absolutely appropriate. I think these data are certainly very promising, but I think on the basis of these data alone, especially because the primary endpoint was investigator-assessed progression-free survival, I don't know that that's quite enough for a regulatory approval. But it really does open up FGFR2 inhibition as a new therapeutic target in esophageal gastric cancers.